You've got trillions and trillions of those little robots in you. They're motor proteins. They're not even alive. They are just macromolecules. They are, I'm sure you will agree, absolutely ignorant. They're marching around, carrying things around in your cells. Not a one of them knows who you are or cares. But think, we know. We know who we are. We care. How is this possible? How do you get from absolutely ignorant, mindless little motor proteins and the like, all the way to the mindful, caring, wonderful, conscious us? That's what the bubble-up path has to explain. One of the most important events in that bubble-up path happened after life had already existed on this planet for about 1 billion years. It happened about 2.5 billion years ago. At this point, all there were simple cells, prokaryotes, bacteria, basically. And then one day, something rather special happened. One prokaryote, A, oh, this would be A, I guess, was invaded by another prokaryote, B. Well, now what? Was A eating B, or was B eating A from the inside? To eat B is to take it apart and use the parts for your own devices. Well, when you're a prokaryote, it's not so easy to tell. On this occasion, A didn't eat B. A pretty much left B alone, but joined forces. It joined forces with the cell, taking advantage of all that design. Now stop and think about that. Here's A, here's B. Each of these have been evolving for 1 billion years. You have 1 billion years of R&D, and they're different. They come together, and the result gets the benefit of a tremendous boost in design work. A whole lot of R&D, a whole lot of extra design is simply added right there. Now, it's very important that you not dismantle B, because that's undoing all that design work. You just get raw materials then, undesigned stuff. The trick is not taking it apart, but reusing it, putting it to new uses. Lynn Margulis, my friend and colleague from UMass, is the scientist who probably did the most to champion this idea of the symbiotic origin of the eukaryotic cell. It's obvious today, when she first pushed it, it wasn't her idea in the first place, but she was the one that championed it. And it's now in the textbooks, and it's clear that this is what happened. And in fact, you can see it in 100 different ways. What you see here, it's not very large on the screen for you. I apologize for that. You see a prokaryote on the left and a eukaryote on the right. Eu, of course, meaning better, really good. And the eukaryotic cells, you see, are more complicated. They just have more moving parts, and those little yellow ovals, those are the mitochondria. And they have their own DNA. And that is the most important clue to the fact that they were originally symbionts that moved in, and then they joined their fates and reproduced together. Every human cell in your body is a eukaryote. You are a eukaryote. And each one of those cells has 2 genomes, more actually. But it's got your nuclear DNA, that's your genome, the human genome, as in the Human Genome Project. And then there's the mitochondrial DNA, which you only get from your mother. And that is directly descended from this invader 2.5 billion years ago. Eukaryotes are more complex than their prokaryotic ancestors. They're more talented, they just have more moving parts. That means they can be more versatile. That means that they can do some pretty amazing things. Now here's a couple of sand castles. You see some sand castles made by some big fancy eukaryotes on the right. And you see a sand castle on the left that is made by single eukaryotic cell, an amoeba. No bacterium is going to make something like that. This is a cell, it doesn't have a nervous system, it's just a single cell. But it is complex enough to make a little home like that. Eukaryotes had the versatility, in other words, that permitted a division of labor, which meant that they could make multicellular life like us, and like just about every visible living thing on the planet are eukaryotes. Make us possible by this division of labor. Thus, as you know, we have stem cells which can turn into liver cells, blood cells, muscle cells, bone cells, brain cells, all these different kinds of cells, because they've got so much complexity in them, because they're eukaryotic. Now here's a eukaryote, a little bit more complicated than an amoeba, and not as complicated as us. It's the caddisfly larva. And here's what it builds to get its food in running water in a stream. This is a food sieve. It's a sort of sandcastle of sorts. And as you see, the water comes in the funnel, the food gets stuck on the screen there that the larva builds, and then the larva goes and scrapes the food up. It's a brilliant design. Really wonderful design. It compares favorably, I would say, with another food catching design that may be familiar to you, and that's a lobster trap. Another way of sieving food from water. What's the difference between those two artifacts? Well, there are reasons for the arrangement of parts in both cases, in the caddis larva's food sieve and in the lobster trap. There's reasons in both cases, and they're even similar. But in the caddis, the reasons are not represented anywhere. Not in the caddis larva's little nervous system, and not in the process of natural selection that track those reasons. Now, I call these not represented anywhere reasons free-floating rationales of evolution, and they are ubiquitous. Every adaptation, every intricate design feature of every species on the planet is the way it is for reasons that have been uncovered or tracked by evolution. But these reasons are not represented anywhere until some clever scientist comes along and figures them out in a process of sort of reverse engineering or interpretation. I want to illustrate this with a few vivid examples, or one in particular, and that's the cuckoo chick. Cuckoos are so-called brood parasites. The cuckoo mother does not build a nest. The cuckoo parents do not build a nest. The mother, when she's ready to lay her egg, finds a host species, different species of bird altogether, finds the nest they've just built, waits for them to lay their eggs. Then when they go off to feed, she darts down, quick, lays her egg in the host nest, and rolls one of the host eggs out. That's just in case the host can count. Clever. Flies away, never to return. The hosts sit on the eggs. Since the cuckoo chick hatches earlier, quicker than the host chick, the first thing it does when it's out of the egg is it tries to push the other eggs of the host species out so it can monopolize the food. I'm going to show you a little clip of this.
The cuckoo's egg looks like the warbler's, and the number of eggs hasn't changed. Everything appears normal, but appearances can be deceiving. Something is not quite right. One chick has hatched way before the others, and it is ejecting the remaining eggs.